Hi, Joel Duff here. Welcome back to my channel. Today I just want to dive into a very recent manuscript. This is a paper or manuscript that hasn't been published yet in a peer-reviewed journal. It's been pushed out onto what's called the biology preprint server, and that just is a place where authors can make their manuscripts available for other scientists to read and potentially comment on, give them feedback before they get peer-reviewed by a particular journal. And, so I'm always kind of perusing, what's out there, what's new. And this particular paper that I'm going to talk about today hit me because I've been thinking about genetic networks. I've been thinking about the origin of information in gene systems. I've been thinking about regulatory networks because in my genetics class, we're about to start talking about gene regulation after we finish up with transcription and translation. So all of these things were on my mind, and I also am always looking for something to maybe mention to students because I like to tell them about current literature, current things that are happening. And so I want to talk about this paper about Arabidopsis, and I titled this Origin of Novel Biosynthetic Pathways Within a Species. And you'll see that I underlined and changed the color of the word within. And that's that's the reason I think that this paper is interesting, is because it looks at the origin of novel biosynthetic pathways. In other words, the origin of novel groups of genes working together and variations of those genes, even within a species, such that one individual might have a different set of genes, a different number or a copy number of genes versus another member of the same species, showing how pliable genetic networks can be. And how much our biosynthetic pathways in some species can change even within a species, much less between different species. And so that's what is interesting, and I think to the scientific community with this paper. And so I want to essentially, I'm going to read this paper. I'm going to read the abstract, a little bit of the introduction, and then we're going to look at some of the figures and explore what we can get out of it. And I'll sort of give I'll take the jargon of the paper and try to explain it in the simplest way that I can, like I would to a sort of a really general biology class, trying to give them a feeling for the depth of what's here while at the same time putting it in perspective of the broader ideas of biology and genetic networks. So we'll have to talk about what a genetic network is as well. I can't just assume you have that knowledge. Okay, so origin of novel biosynthetic pathways within a species coming up. Okay, watch out. Here we go. These titles aren't going to grab you. I mean, these are not made to be titles for clickbait. These are titles to inform a scientifically literate audience exactly the type of thing that they're going to try to communicate in this article. And so let me read you the title. Copy number variations shape the structural diversity of Arabidopsis metabolic gene clusters and are associated with the climatic gradient. A lot of words to unpack in there. But before I get to that, I just want to note that the authors here, Marszalek, Cesnak, I don't know how to pronounce these names, et al., I'll say, all right, a group of people. This is a large body of data that has been gathered and analyzed, and so a number of people involved and are all recognized being authors on the paper. And they come from a number of different institutions. You'll see we've got several different institutions in Poland. And I thought most notably, I would just point out that one of the members on this paper is from the National University in Kiev, Ukraine. And as I noted before, this is in the biology preprint server, and this was posted on September 5th, 2022. Okay. Copy number variation. We're going to see this a number of times. CNV, copy number variation. And it is what it sounds like it is. If you have a particular gene in a genome, sometimes there can be one copy of that gene, but other times there might be more than one copy of that gene. Sometimes those copies are virtually identical to one another and have the same responsibility or do the same thing, make the same structure. Other times there's slight variants of each other, and they either function in the same biosynthetic pathway but maybe with different efficiencies, or potentially, and more intriguingly, some of the variants might actually take an alternative biosynthetic pathway and are involved in the biosynthesis of slightly different molecules, resulting in potentially different phenotypes of the organism, a phenotype being an expression of a characteristic being different in that organism. So that's a key portion of this paper, copy number variations. And they're saying these copy number variations shape the structural diversity, all right, the structural diversity, the shape of the genome, the characteristics of the order of the genes, the number of genes, and that shapes the diversity in Arabidopsis. Arabidopsis is this type of mustard plant. It's a common weed in Europe and Asia, and has since spread really all around the world, so it's nearly cosmopolitan. And it is considered a model plant or model organism in the meaning that you'll find Arabidopsis in thousands of labs. It's one of the most widely studied plant, and we know so much about it, which allows for interesting comparisons, and we know a huge amount about its biochemistry. All right, so that's the plant that we're going to look at. And then we have this phrase, metabolic gene clusters. And we're going to see that as MGCs. I can't write today. MGCs, metabolic gene clusters. And those metabolic gene clusters, which I'll give you an example in a moment, are supposedly, or they find evidence they're associated with climatic gradients. And I'll just give away the part of the story there because I'm not going to spend a lot of time on that. And that is, they find that there are different biosynthetic pathways in different individuals within the species of Arabidopsis, the commonly studied Arabidopsis. And there's a correlation between the copy number variations, the number of copies of certain genes in these gene clusters. There's a correlation between the numbers of copies and the climatic gradient. The climatic gradient would be if you move from the ocean up into the mountains, right? You're going to experience different climates. And these plants grow in a wide variety of climates, but if you find plants that grow at higher elevations, cooler temperatures typically, maybe wetter, you find that they have a different genomic structure than others that live at other elevations or other climates. So the genetics seems to follow a climatic gradient, suggesting that the environment is working to shape the organism and adapt it for those different locations. All right, let's get into it. And in order to really get into it, we need to discuss this idea of a metabolic gene cluster. And so to simplify it here, we have a diagram of let's say this is a portion of a chromosome, all right? And we're talking about chromosomes. Chromosomes are actually proteins, DNA wound around proteins, but we'll just consider the DNA. Most people think of the chromosomes as being the hereditary material, the DNA portion of it. And so a string of DNA, a string of code, A, T, C's, and G's, and on that string of DNA, there are particular portions, say from here to here, and here to here, and here to here, all right, which have a certain amount of code. So let's say there's 900 A, T, C's, and G's, or nucleotides. All right, so 900 nucleotides makes up the code for that particular, what will eventually be a protein. And then there's another, and we would call that a gene, right? So this is a gene right here. And that gene is read and translated into a protein. And that protein then, in this case, we're talking about enzymes, because we're talking about metabolic gene clusters, which are clusters of genes. Okay, so you have gene number 1, 2, 3, 4, maybe 5. So we have 5 genes here. And the 5 genes are making 5 products. The products are each an enzyme. So enzymes are proteins that are folded up amino acids. And those 5 different enzymes, each enzyme is responsible for catalyzing or helping a chemical reaction occur. And so here we have an enzyme over here, and there's some kind of precursor product over here in the organism, some kind of molecule, right? In this case, we're showing it as a ringed, a 6-carbon ring probably. And this makes this 6-carbon ring. And then the next enzyme has the responsibility of tweaking that particular biomolecule. And so they're showing, okay, let's add in something here. And then this enzyme catalyzes or helps the chemical reaction occur that then adds something else to it, right? And so what are we doing? We're building a more complex molecule, and it has to go through a series of steps in order to create this complex molecule. The steps are helped along or governed by these different enzymes, and each enzyme comes from a gene, and that's where we can say that your genome is full of lots and lots and lots of different pieces of code, right? Individual genes that make enzymes. Enzymes are responsible for constructing things that are the physical structures, right, in our bodies. And so the enzymes then are doing making or synthesizing things. And so that's this metabolic pathway. It's a pathway of making this end product, and say this end product is the thing that once you put all these pieces together, that thing will then have some responsibility, right? It's doing something. Maybe it's an actual structural component, but it could also itself be a part of some important process like cellular respiration, right? Or, making a piece of protein, a piece of your muscle or something like that, right? And so metabolic gene clusters are just the idea that there can be groupings of genes that when they're expressed, they're working together to do one pathway or one metabolic process. Now, interestingly, this is what happens in bacteria a lot. Bacteria have something called operons, and operons are groups of genes that all work together. In fact, they're all copied together all at the same time. They're very closely linked together, even more closely than shown here. And in eukaryotes, and you and I and plants and fungi are eukaryotes, we have a nucleus and we have a far more complicated genome. You still might think that it might be useful to have a bunch of genes that are all involved in the same biosynthetic pathway, right? The same thing you need to make, and you need lots of things to help do it. You would think it would be convenient to have all those genes all together in one part of your chromosomes. And so you could easily access all of them at the same time, and then boom, I need to make this product, right? And so I need to make enzyme X, Y, and Z to do that. You would think you'd have those all together, but in fact, the eukaryotic chromosome often has these genes sort of all over the place, right? Gene number 1 could be on chromosome number 1, and gene number 2 might be on chromosome number 3. Gene number 3 might also be on chromosome number 3, but be a long way away, and there's another gene in between it. And so when you want to do this biosynthetic pathway, you have to go find all those pieces, right? You have to go find each one of those genes, turn them on or express them and make the enzymes, and then the enzymes would then be in the cells, and then they would guide the chemical reaction to occur to make the final end product that you're seeking to make. And so we tend to actually spread out our genes that are doing these metabolic functions rather than clustering them all together. You can kind of think of it as your hard drive. You might think that when you have a program, then you start your executable file. You can think of that as gene number 1 here. You start your executable file, and it has the information for, like, you need to go and upload all these other files in order for you to have all these files running and being active in order to have this program run. And you might think that all those files are just right next to each other on the hard drive, but in fact, sometimes even parts of one individual file, which you might think of as a gene, some parts of that single file might actually be separated into little pieces around on your platter, right, on your hard drive. And they may not all be collected together, but you have the information to go find all those pieces, put them together, and then run that particular program. That's kind of what a eukaryotic genome is doing. All right, so now I'm talking a lot about this, but I'm kind of giving you a flavor for the genomic structure of things. And so what's interesting about metabolic gene clusters in, say, Arabidopsis is a plant. There are some areas in the genome where a group of genes, a small group of genes, end up being together, and they all work with the same, and they all participate in the same metabolic pathway. All right, so they are a biosynthetic pathway set of genes, and they are clustered together, kind of like in bacteria. Each one of them can be turned on separately, but at least they're next to each other, and so that can have advantages for having them all expressed at the same time. All right, so that's the idea of metabolic gene clusters. And in Arabidopsis, this paper is going to go out and say, we're going to look at a bunch. Well, they found 4 different clusters of genes. And then what they really were interested in is the gene cluster always the same in different individuals? How much variation are there in gene clusters within Arabidopsis? And so let's just go right to the abstract. Let me read the abstract and interpret it for you. Then we'll read the introduction, which will give us an idea of why what they're doing is important for the rest of the scientific community or genomicists, and even evolutionary biologists who are interested in how do genomes change and how do they adapt. And what I think that what these authors don't talk about much in this paper, and they don't emphasize, but what they're really doing is they're demonstrating how new genetic information can arise within a genome, how new biosynthetic pathways can find their way into genomes by adapting, copying genes, and finding new functions for them. So we'll take a look at that in a few moments. That's when we get to the figures. I'll be able to illustrate that. So first, abstract. Background information. Metabolic gene clusters, so there's your MGCs, encode, right? They have the code in the DNA, at least 3 different enzymes for a common biosynthetic pathway. So for us to call something a cluster, it has to be more than 2 genes that are in the same vicinity. So at least 3 different enzymes, and sometimes 4 or 5 or maybe more. So that's what a metabolic gene cluster is. Comparative genome analysis highlighted the role of duplications, deletions, and rearrangements in multiple gene cluster formation. How do gene clusters arise is what they're trying to answer here. Where do they come from? Why are there clusters? In eukaryotes, typically there aren't clusters of genes, of enzymes that are part of the same biosynthetic pathway. So when you do find a few of them, you wonder, why do these exist? And so this sentence is emphasizing the comparative genome analysis. That's where you would take genome, you take a genome from one organism, genome from another organism, genome from another organism. It might be different species, it might be individuals within a species, and you line up those genomes and you look at the order of the genes and where the genes are located. When they look and find gene clusters, they have identified that those gene clusters often have duplications, where one gene might be found in 2 copies in one organism or one individual, but only 1 copy in another one. So that could either be a duplication, or depending on whether that was the original state where there's 2 copies, it could be a deletion in one other individual where one of them lost it. All right, so you have duplications and deletions relative to the ancestor of the original state of that particular genome. And then also rearrangements where the genes get moved around. They get turned around such that they swap places with other genes, and that can affect potentially the biosynthetic pathway and well, I guess you could say it's going to change how the genes might be regulated, how they're turned up and down in terms of the volume or the amount of expression they have, which is going to affect how fast maybe a chemical reaction occurs. All right, we're through 2 sentences. We hypothesize that these mechanisms, duplications, deletions, and rearrangements, also might contribute to metabolic gene cluster interspecies diversity and play a role in adaptation. So the previous sentence, what they're really saying is people who have compared different species or maybe different genera, like really different organisms, they've seen that where new genes come from and how big changes in the genome occur are duplications of portions of the genomes, rearrangements of those genes, deletions of genes, forcing the organism to change and switch their biosynthetic pathways. That's sort of big differences between organisms. But we hypothesized what we're going to do here is we're going to ask, are those same mechanisms occurring within a species, just between different individuals within a single species? Do those mechanisms also contribute to diversity within species? That's what intraspecies means. And therefore possibly play a role in adaptation. So that's where the climatic gradient comes in. They're going to say it's not just random. We don't just have random changes. We have changes that then seem to be sculpted by natural selection. In other words, certain changes were selected in different environments. And so these new arrangements of the genes, creating new biosynthetic pathways where they're making slightly different products, or maybe they're turned on differently in different tissues at different times in different places because of their rearrangement. That's resulting in their being adapted to different climatic zones. And so these changes in the genome are allowing the organisms to then adapt to their different environments. All right, what are their results? We assessed copy number variation, CNVs, of 4 Arabidopsis thaliani. That's the particular species of Arabidopsis. There's actually other species of Arabidopsis, by the way. But Thaliana is the one that's most studied. Multiple gene clusters in a population of 1,152 accessions. All right, so you can think of that as they have 1,000 genomes. You know, this still blows me away, even today. When I was in graduate school in the 90s, I couldn't even imagine having one genome to look at from an organism, much less hundreds or thousands of genomes of a single species in order to compare. So we can get to such fine level detail of how organisms or how genomes are changing. We're fortunate to just be able to look at 1 or 2 genes and maybe look at a few individuals for those genes, much less the tens of thousands of genes in an organism, and look at their entire organization of those genes in the genomes. The multiple gene cluster diversity was lowest in, and then he goes, this is going to go on. It's going to say there's this certain gene cluster, and we found a lot of diversity here. There's another gene cluster, and we found that some individuals had a gene, and some other individuals in the population were missing that particular gene in that cluster. So the cluster was actually different in some individuals than others. But I don't think I need to read that. I'll show you some pictures in order to visualize this. But that's the middle of their abstract. It's just outlining, here's our results. We found this in these types of ratios of different genes and gene clusters. Conclusions. Our study highlights the importance of interspecies variation and non-reference genome. So the reference genome would be like the genome of the we'll call it the domesticated Arabidopsis that's in labs. That comes from a small number of individuals from the wild, and that sequence is known and has been known for a long time. And so what they're doing is they're going out in the wild, and they've actually collected or gotten other people's sequences from other wild populations to try to get a feel for the total diversity of all Arabidopsis genomes around the world, to see like how much diversity is there within this species. Back to conclusions. Our study highlights the importance of interspecies variation in non-reference genomes for dissecting secondary metabolite biosynthetic pathways and understanding their role in adaptation and evolution. Now, the secondary metabolite biosynthesis, secondary metabolites, for the sake of what we're talking about here, you can think of these as more minor products that plants are using. So maybe defensive chemicals that they use in order to stop insects eating them. So if you create a whole bunch of toxic chemical, then your leaves are going to be less palatable, right? And so that prevents as many insects from chewing up your leaves, which then protects the plant. So they're a form of, I guess you could say it's a form of immune system, sort of. There's all kinds of plants are just amazing in terms of the number of total chemicals they produce. They have so many different biosynthetic pathways. And some biosynthetic pathways produce things that are absolutely essential for life. And then there are other biochemical pathways that are producing a wide variety of like a whole mixture of different chemicals that that might collectively be important to the organism, but losing one of them is probably not going to be like lethal to the organism. And that's important to remember. That, and this is, if you think about this now, the reason this is important is because there are certain biochemical pathways or biosynthetic pathways where if you tweak it a little bit and you make a slightly different chemical, so you make a different product because of the changes you made, like you lost a gene in the pathway, now it has to skip a step, or you just tweaked one of them because you had a mutation and now it changes the shape of that enzyme so it functions a little differently. You're not going to kill the organism. You simply make a different biosynthetic molecule. So you're making a different pigment, or you're making a different, something that is of, we'll call it minor importance, but potentially has some adaptive value. And so potentially could be in the world of natural selection where you're living in different environments. Oh, well, this change that I just experienced that changes this biosynthetic pathway because my genes got rearranged, or I lost a gene, now suddenly allows me to make a new chemical that in a cold environment gives me some advantages over my neighbor, which can't do that. And so this is how new biosynthetic pathways might arise and begin to be used by certain individuals within a population versus other individuals in a population living in a different environment. All right, but I get slightly ahead of myself here. Let's go to the introduction. And so let's get a little more flavor for this idea of multiple gene clusters. Plant MGCs are thought to have risen by duplication and subsequent neo or subfunctionalization of genes involved in primary metabolism, which might have been followed by the recruitment of additional genes to the newly formed biosynthetic pathway. Whew, okay, let's dig into that for a second here. All right, so plant multiple gene clusters. So here you have a multiple gene cluster. It's got 4 or 5 genes. Well, we're back to the original question. Where did this cluster come from? How did it become a cluster? All right, so it might have arisen by duplication, right? So some of those genes, sometimes what you get is if you're working on making a particular biosynthetic pathway, is probably making slight variations of a particular organic molecule, right? You're like, I got this organic molecule and I want to make a little tweak here. I want to add a hydrogen over here, right? And I want to add an OH group over here. And the enzymes you use to do those are probably somewhat similar to one another. They each are slightly different from each other and have these different jobs, and they kind of sequentially work on one after another. So one way you can diversify the number of different ways you can tweak a molecule, right? Tweak one of your organic molecules that you have in your body, is you could duplicate that gene, right? And if you happen to have a gene duplication, which is typically just an accident that happens where you copy the gene, so now you have 2 copies of the gene, and you have both of them on, well, one thing that could happen there is you can make twice as much of the enzyme, in which case you're essentially speeding up the chemical reaction. So that's an actual change in the expression of the genes, which means you might be able to make more of that product. And so maybe you're creating more enzyme, which means you're doing more biosynthesis, which means you're making more product, and that might be an advantage in that environment. Or it might be a disadvantage, right? Either way, it's a change in the phenotype. It's a change in how these products are going to be expressed in an organism. Well, now, okay, all we've done is gotten halfway through that sentence. We're to arisen by duplication and subsequent neo or new or subfunctionalization. So subfunctionalization would be, hey, I got one gene, I copied it, and now it forms a subfunction, like I have a mutation in one copy that's not in the other, and now I have a variant there, and that variant can do a slightly different biosynthesis than the other one. And that helps me do something just a little bit different. Neo means sometimes if you create a duplicate and then you have a mutation that is more radical. More radical means it changes the amino acid structure more, and so you end up with a very different enzyme that isn't really very close to the catalytic function of the other enzyme. Well, then it's going to catabolize something else, in which case you might have just created a gene that will potentially help out another biosynthetic pathway, not even the one you were originally part of, but you're in the cell and you're now making this new product, and then somewhere else there's another biosynthetic pathway where if you add in this other enzyme, maybe it allows you to make two variants of this organic molecule instead of the one variant you were making before, and then you can there's a way that you modify that one because of some other enzymes that are already part of another biosynthetic pathway. In other words, what happens is your gene changes its responsibility now from working with one biosynthetic pathway, and all of a sudden it's off working and helping out another biosynthetic pathway. That would be more neo because it feels more new, right? It's like I have a new function. I'm not even like really related to the old function. I'm doing something brand new. So you're either tweaking or potentially radically changing the function. All right, to continue. So that's functionalization of genes involved in primary metabolism, which might have been followed by the recruitment of additional genes to newly formed biosynthetic pathways. So I'm taking some tangents as I'm talking through this one sentence. So to work back, multiple gene clusters, how do they originally arise? Well, you might have had one gene that then copied itself, and now it's able to do two parts of a biosynthetic pathway. And then there was some other gene that now is, somewhere else in the genome, maybe 100,000 base pairs downstream. And down there, it's also making a product which actually is useful in that same biosynthetic pathway. And so that gene and one other gene, another gene that are making similar enzymes and tweaking the similar biomolecules, they create a new biosynthetic pathway. All right, so now you're making a new end product because you've now combined 4 or 5 different genes that are kind of working together. But they're not necessarily next to each other. In other words, they're not a gene cluster. They're still separated, just like most of our genes are in biosynthetic pathways. But if there's an advantage to them being closer together, and you have constant mutations happening in genomes in these plants, you have inversions, you have things where they're moving sections of the genome around, and you end up with 2 of those genes being closer together. And we talk about cluster, and we showed a picture. They look like they're really close, but you have to understand sometimes 4 genes that are next to each other still span an area of 100,000 or more base pairs. It's still a large area in the genome. And so now they're in the vicinity of these other genes. And as I said before, there can be advantages to how you regulate genes in that particular vicinity of the chromosome, such that now you can kind of you can control all of them kind of together. And that would then make that particular biosynthetic pathway a little more cohesive. It's sort of like, okay, the plant's like, under this condition, I'm living in a cold environment now where it got really cold. I need this antifreeze molecule. And if I just upregulate this beginning portion of this cluster, it will also affect all the other members of this gene cluster, which then all begin to express themselves at higher rates, and I start to make more of this antifreeze. And that'll give me an advantage in this particular environment. So now what happens is that biosynthetic pathway has become more established in the genome. At first it was just, here's some genes out there. Some of those genes have some other responsibilities already. They're making an enzyme that's part of another biosynthetic pathway. But you have to appreciate that some enzymes are actually involved in multiple biosynthetic pathways. They're not just one thing and they do one thing and they're never involved in any other chemical reactions. Some of these ones can do multiple things. And so what you're doing is you're creating new networks, new pathways. And the more you can link them together And the tighter you can kind of cluster them together, the more likely you're going to be able to control them together. And the plant over time is going to be able to then get more and more consistent use out of those particular pathways. And so what I'm trying to illustrate here is how you can go from a fragmented network of different genes that are doing other things and eventually knit together a new combination of genes. But it's not just taking old information and then reapplying it in new ways. I'm going to show you in a moment that you actually have genes that have duplicated and then they've taken on completely new roles within that biosynthetic pathway, allowing for new functions to arise within a genome. And the amazing thing here is this isn't just making new species. This is making new genetic information and new characteristics, new traits within one species. So we're not really dealing with. We are dealing with speciation because ultimately this might cause speciation to occur within Arabidopsis, such that the one species finally divides into multiple species or two species, because they accrue so many different genetic pathways between different individuals that eventually, once they're growing in different locations, accrue so many differences, they become more isolated from one another. Let's keep reading here. Now, here's an interesting observation they made, and this is the power of being able to look at 1,000 genomes and being able to look at patterns. They looked at 4 multiple gene clusters that they found in Arabidopsis. So by looking at hundreds of different genomes, they found 4 clusters that are typically found together, sets of genes that are together. And where they found them was interesting. They found them located within dynamic chromosomal regions. Dynamic chromosomal regions are regions that experience a greater pace of change, of mutation, of rearrangements. So, for example, subtelomeric, centromeric, that's at the, at the center of the chromosomes, that's where they fuse together. When 2 chromosomes copy each other, that's where they're fused together at the centromere. Places that are rich in transposable elements. So transposable elements are those jumping genes, right? Things that are moving around, copying themselves. Well, what happens when transposable elements move around? They also make mistakes, and they also copy information that might be near them and duplicate that information. So this is how you get duplicated genes. You get duplicated genes because they're in active regions of genome reorganization and information being swapped in and out. And so you have a high rate of stuff moving around. This gives many opportunities for finding new pathways. So this is happening in multiple different places, and most of the time these don't really have any effect. But once in a while, it's going to be through the random nature of this, you're eventually going to sometimes bring genes together that can actually find some way to work together to form a new pathway that might not have existed before and form these multiple gene clusters. The possibility of bringing together beneficial sets of genes by recombination, translocation, moving from one place to another, copy number changes, so accidents of copying, and rearrangements may be higher in these regions than the rest of the genome, thus promoting multiple gene cluster formation. However, the same factors can contribute to the accumulation of genetic changes in already preformed multiple gene clusters. So even where there's already a gene cluster, if it remains in an area that's highly active in a genome and experiences a lot of change, then that gene cluster can either get disrupted and broken and lost in time if it's not so important to the organism that it's going to cause them to be selected against. Or it could actually continue to increase the importance of that particular gene cluster because it can add even more genes to it, making it more powerful because it might find other biosynthetic pathways to explore, and therefore it's going to go from doing one little job, making one final product, to maybe, hey, we add a couple more genes here, tweak a few things here. Pretty soon we're making 4 or 5 different products, right? We're making 4 or 5 different variations of this chemical that is a defensive chemical that now gives us even more defense against being eaten by some herbivore. All right, so they hypothesized that that they need to look in these locations, look for the action of those things, and then, all right, to verify our hypothesis, we evaluated the structural variation of Arabidopsis thaliana through a combination of bioinformatic approaches and so forth. And then they go on and on and on about, like, okay, here's all the things we did. Well, what did they find, right? We don't need to go through every single step. We just need to look at some examples, because what I'm trying to just illustrate here is that there is a really amazing amount of variation in the structure of a genome, even within a species. And so there are vast portions of the genome where almost every member of the species will have the same genes and the same order, and they might have some slight differences in their sequence, different alleles. But for the most part, there's not going to be a lot of change. There's going to be a lot of stability in most of the genome. After all, if there was flexibility everywhere in the genome, and rearrangements and transposable elements were flying all over the place everywhere, the genome would be degrading in the sense that it wouldn't be able to maintain its function very well, right? Because most of these things are going to cause problems, not help things. But the vast majority of the genome is stable. It doesn't change rapidly. And generally, that's the core systems, right? The core operating system in an organism, the most important gene pathways, the most important biosynthetic pathways that you have to have, and you can't play around with or you'll be dead. Those are in more stable portions of the genome. And where the organisms are playing, all right, are on the fringes. It's like, these other lesser important biosynthetic pathways that are making chemicals that we could either have or maybe not have. And, that's where we're kind of like, we're in this area where we can play. So now let me show you one of these. Oh, these are the 4, by the way. So they looked at 4 different multiple gene clusters. Something that makes this product, I can't even pronounce these things, thalianol and marinal and this and this. All right, there's your 4 different biosynthetic pathways. Those are the names of the pathways. And then the colored genes in here, all right, the ones that have a particular color like this orange, those are all the genes that are involved in that particular pathway. And then there's other genes too. Here's a gray one here. Here's a gray one here. And those genes are actually genes that are not involved in that pathway. So these multiple gene clusters aren't, I showed you a simplified, cartoon, right? Where it's just like, here's 5 genes, they're all next to each other. A cluster is still just like, hey, there's a group of genes in this area, but there's still other genes in there. And that actually lends support to the idea that this is an area where things are changing in the genome a lot. Things are moving around, and they've managed to kind of get these genes in the same area, and they work efficiently. So maybe natural selection is saying like, hey, that was a good combination you just made there. Let's kind of go with that combination. But we still have these other genes that are functioning in that region too. We need to keep them as well. So there are other genes mixed in here as well. But the idea is that generally you still have genes associated with one another in terms of making products, working together in a biosynthetic pathway. So these are the 4 different, and what they're showing here is just your most common form. All right, if you sequenced 1,000 genomes, 1,000 individuals, a lot of them have this particular structure. A lot of them have this particular structure. That means this is where the genes are. This is the direction they're copied, and so forth. This over here, which I know you can't read these things, but all you have to know is that these represent individual genomes. And they're looking at this particular pathway. And there are individuals that have either don't have a particular gene, right? See, some have this particular gene. The genes are listed down here. Some don't have it, and some have that gene in their cluster. And then other ones have this other gene in the cluster, but they don't have this other gene in the cluster. And other ones have yet a different gene that's unique to that particular cluster, or found in a different combination. And so just looking at this, you can get an idea that, yes, there are individuals, there are members of populations that have different structures to this biosynthetic pathway. So let's look at one of those. All right, so here's one of them. I can't remember which one this is. I think it's the Marnaral. And again, we're back to kind of a simple diagram, but it gives you the idea of here are the important genes that are part of this gene cluster. And here are the really clever names for those. Okay, all right. And so here we have reference, which would be like, here's your typical Arabidopsis, so our lab variety. And we've sequenced that genome a long time ago, and it has like this gene, this gene, this gene, this gene, this gene. That's the order of those genes. Look, there's 1,000 of these things that have this particular gene order. And then we started looking out in other populations and other individuals in the wild. And we find there's 37 different genomes that they looked at, and those genomes have this gene is present, but these two genes are missing, and then this gene is also missing, right? It only has 2 genes as part of that biosynthetic pathway. Now, the other thing is that they did, and this is what makes this isn't just a, sit back with your computer and do a genome comparison study, which you can do, and there's thousands of publications that are done this way. They took the extra step in some cases of saying, if you go look at that individual, are they still able to make like some kind of product there? Does missing those genes matter? In some cases, it does matter. It does change the biosynthetic pathway. In other cases, there's an extra gene there. Well, like say, for example, right down here, we've got an extra copy, right? This is not much different from this one. It just has 2 copies of this particular gene. And there's 5 different individuals that had that. And what they found is that, it actually still functions and still makes the same product that the other one does. Other ones might disrupt the function, but in many cases, they make a slightly different product because they have a different combination of enzymes, and they kind of like, I can do this and I can add this and then I'll add this, but I can't add that, right? And so not all biosynthetic pathways are just a linear thing where you have to go down the line. There are things where you can skip steps potentially, and you can end up with a different product. And over here, this is showing there's something else that was going on in these, which is not shown in this picture here. But there is one area where some individuals have genes. This black bar, you have these 2 here, and then the whole thing is flipped around, and so the genes are facing a different direction, so they're in different order, right? So that's a rearrangement. And the rearrangement may or may not have anything to do with the final function or the final product that's made by those, but rearrangements can change the way that genes are regulated and therefore change the overall expression. And so they looked at these different basic genetic groups and different genotypes, because overall these are different gene structures in different individuals. And they found that across the world, there's areas of the world that have more of one type than another. And so we can see that the populations are changing. And through the paper, then they're going to try to show that this isn't just random. It turns out there is, I said before, there's a correlation between climatic zone and where you find particular combinations of genes, suggesting that different combinations of different gene structures for the genome may have different phenotypes, and those phenotypes are being selected by the environment, right? So their rearrangements and their duplications are resulting in phenotypic expressed changes that the environment then is selecting as either that was an advantage or a disadvantage in that particular environment. And so some individuals are being selected for, some are being selected against if they have that particular combination of genes. So let's go on to another one. This is another gene cluster of the 4. This one's a little bit more complicated. And what we really care about is the PEN1, BARS1, and then there's BARS2. And you'll see that even the size of these genes has changed. So this gene makes an enzyme, but down here we see that this gene is even larger, and these are all different particular strains of Arabidopsis, so individuals of Arabidopsis. And what you're seeing is a map of their genome. And for the most part, look at these first couple. It's like, hey, we got this gene, then we have this gene that's being read in this direction, then we have a gene being read in this direction, we have this little small gene here, then we have this PN1 gene. Then we have two other genes that aren't really part of this biosynthetic pathway, but this BIRS gene is part of our biosynthetic pathway. And then down here, they had this little triangle. And what the triangle represents is if you go right down through here, let me see if I can get this right. You'll see that there's a BARS2 right here. And the BARS2, look at this purple gene right here. And then look at that gene and the next two. Actually, I might not be doing this right. Anyway, this whole thing here is almost the same as this right here. Right? Whoa. That right there. Oh, what a mess. All right. Anyway, BARS2 represents a copy of BARS1. All right, so BARS2 is a copy of BARS1. And it's thought that somewhere in time, this is based on comparisons with other Arabidopsis, by the way, because other species have this structure. And then there are some species that, oh, I know, this green one right here, you'll see the green one's over here, right? So what you have is you have this big section here. This whole section right here is an insert. And that's what this triangle's for, saying there's a big chunk of DNA inserted here. You know, there was a population of Arabidopsis, maybe the whole species at one time, that only had this particular genome structure. And then sometime there was a duplication of these 5 genes. And those 5 genes got copied again, tandemly it's called, so you got added on. And then since then, some of those genes have changed. So sometimes some individuals have lost, like this one, it's missing one of the genes. And other ones have a gene that's turned around in the opposite direction, I think. All right, so there's been an inversion there. So since then, you could say that the genomes have been evolving in other individuals as they descend and make copies. They've been making further errors and changing this portion of the genome. Now you'll see that they haven't really changed anything about the original core set that was the same as the original combination. But over here, they've changed quite a bit. What's going on there? Well, what happens is if after they had the duplication event, these can continue doing exactly the job they were doing before. And so maybe some of these other genes were important for something, in which case, if they're important, you can't really play around with them. You can't just lose them. But then after they've been copied, it's like, well, wait a second. I already have, where did I do this? This one right here, right? It's like, I already have a copy of this one. It's already doing it. The plant's surviving fine because it already has a copy and it's being expressed and it makes whatever it makes. So I don't need that one anymore. So if I have an accident where I lose it, well, it's just lost. All right, that's a deletion. All right. But what about this BARS2, which still exists, and none of them have been lost? If you go down here, this is showing how different they are genetically. All right, so this is a phylogeny showing that all the BARS1 sequences, that would be all of these, they're all genetically very similar at the DNA sequence level. This is like you sequence the DNA. It might have been sequencing a protein. I can't remember if this is a protein phylogeny or DNA phylogeny. Either way, what this shows is that every BARS1 from all the individuals that are looked at across these populations across the world, their sequences are all really similar to each other. That suggests that gene has an important function. It makes a particular enzyme. That enzyme needs to work a certain way. It doesn't fool around with that gene very much. And shows they're all related by common ancestry, right? They all have a common root. What about BARS2? Well, BARS2 has a long line going from it to BARS1 saying it's actually changed a lot. So after the duplication happened, the duplication might have happened here, right? And after that duplication, you have BARS1 continues to change, and then all these are the living individuals. All the living individuals are related to something not too far in the past. But you go farther in the past, you had this duplication event, and that experienced a whole bunch of changes to all the members that are in the present. And the present ones also are very similar to each other. Well, guess what? They have a very different DNA sequence, but not so different that you can't tell this is essentially the same gene, except that it has enough differences that it biochemically does something a little bit different. And in fact, they've tracked that down to members that have the BARS1. Now let's see. Oh boy, I'm going to have to think about this now. I don't have it right in front of me. I think it was BARS1. If you have the BARS1 gene and this biosynthetic pathway, this biosynthetic pathway is expressed in, I think it's the roots. So it makes a product in the root cells. But it doesn't, these genes don't turn on, they don't do anything in the shoot, like up in the leaves. But the individuals that have BARS2 and have this stuff, apparently if you have the BARS2, when it turns on, it actually is regulated in such a way that it turns on in the shoots. And then it uses or somehow influences the rest of the biosynthetic pathway to be turned on. So you're using those other genes and you synthesize this product in the leaves. So before you only did it in the roots, but by copying the gene, you now allowed a new regulatory thing to happen. So gene regulation is a whole nother added thing on top of this. But the presence of this additional gene allows you to regulate it differently. Before you couldn't really play around because you needed to express in the roots, so you couldn't change the regulation there so that it would express in two different places because you really needed it mostly in the roots all the time. So, but now having an extra copy allows you to hey, you know what, with this extra copy, we can regulate it differently than we did the other one. So this one apparently keeps the same function. How do we know this one is keeping the original function? It's because it's like the same sequence, all right? It's making the same enzyme. It's part of the same biosynthetic pathway as all the other ones that don't have BARS2. All right, so I go through all of this to show that this is within a single species. You have one individual that has 4 or 5 extra genes, right? But they're copies of other genes they already have. And that's originally was a mistake, but this organism has found a way to use that mistake to now add a new function, because the plants that can express this in the leaves have a function that the other plant does not have. Right? It doesn't have it. It can't do it. And so you have added, I'll call it, new information to the organism, right? I mean, how could it be anything else? It's a new piece of information that allows you to do a new thing for that particular organism and express that thing in a different way. All right, there's other examples in this paper, but this was like the clearest one, so I picked that one out to show, because I'm not going to go through all 4 of these gene clusters. But all 4 of the gene clusters show variations, and it's thought that most of the variations probably have some kind of actual physiological effects on the organisms. All right, so according to our current understanding of MGC formation phenomena, non-random gene clustering in eukaryotes is linked with the highly dynamic chromosome regions. All right, so where we don't find randomness, because most genes are kind of look like they're randomly thrown around the genome, where we find these clusters seem to be associated with dynamic chromosomal regions where you can have lots of changes and movements and reshuffling and duplications. And at the same time, then you're going to have selection in that environment in those individuals that is going to select for which combinations, which duplications, which changes in the genes work in that particular environment. So what individuals will be the survivors of those events? What else did I want to get out of here? All right, so then go on to say presence and absence, variations, tandems, and segmental gene duplications, recombination, gene shuffling, and transposable element mediated variations are considered as the main genetic drivers of metabolic pathway profile diversity and multiple gene cluster evolution in plants. Now they go on to say, look, this probably is also true not just for Arabidopsis, because that's what most scientists are trying to do, is find patterns that don't just work for one individual, right, or one species. They want to say, is this something that could happen elsewhere? Recently, population scale studies highlighted the importance of intraspecies, within species genetic variation, elucidating the role of these factors in the formation of gene clusters. Resequencing of genomes of 9 opium poppy accessions with a range of alkaloid production profiles revealed copy number variants affecting the majority of known benzoyl isoquinol alkaloid pathway-related genes, and they're paralogs. Paralogs are copies of genes that now have variations in themselves and are possibly expressed in different ways, but they're considered paralogs or related by common ancestry to the same gene. So they're genes that have duplicated and taken on new roles. So it's kind of like gene phylogeny, right? The genes that have evolved into new functions. Pathway genes located within clusters were about 3 times more likely to exhibit copy number variation than those outside clusters. So if you look at genes in more stable portions of the genome, and often those genes are involved in more centrally important pathways or things that an organism does, are much less likely to have these gene duplications. All right, and this makes sense. You want to protect the core things that you do that are really essential, but you also want to be able to adapt to your environment. And the way to adapt is to change things on the periphery of the things that aren't quite as important. And you can dink and play around with those and do these adaptations to the environment that way. But that ends that paper. But I want to draw out something else. And I've been kind of saying that throughout my talk here. But I'm going to try to illustrate it in 2 other ways. Because this gets to the question of, like, how do you develop new characteristics? Like, this comes up in the class all the time when you're talking about genes and genetic pathways, and how do you get such a complicated pathway to making a particular product, especially when a pathway is like a developmental pathway for, like, how do you make an eyeball or something like that? How do you get all those genes coordinated with each other so that they do things in the right order? So what you're seeing right here is just and you don't have to you don't have to know what any of these different gene names are and what they do, and you don't have to be able to see them. You should just see that this is a genetic network analysis showing association between significantly modified or modulated genes, and the arrows are just pointing to this gene affects this thing, which affects this thing, and this thing affects that, and this affects that, and this gene does this to that. This really essential gene is actually affecting multiple different things. So if this gene comes on, is turned on and expressed, it will actually affect a bunch of other different pathways. So here's the thing about this. There are certain genes in here, like especially these ones that are in orange, that kind of are like central, right? Is like if this gene is present and active, it's affecting multiple different pathways, multiple different things. So this thing is like affecting that, but also affecting that, but also affecting something else in another cell system. It might affect something else. These are really core important genes then. And they're really harder to change, because if you change that, you might change 5 pathways. If you take one of those out and you delete one of those genes that's at the center of a genetic pathway, well, then you're going to end up losing a whole bunch of things. That's probably going to be lethal. But what if you have a mutation to, I don't know, MEF2B right out here, right? It's only connected to one thing, which is part of what, I don't know. Maybe this is not the most important thing that any organism is doing, and it has some function that is useful. But if you lost it, you might not really even know it. Okay, there are systems like that, and so effectively, these genes out here are more pliable. They're more changeable. You can have mutations there. You can have more mutations. And when you make mutations, here's what can happen. This, I got to move over here because I've done too much coloring over here. This gene right over here, doesn't look terribly important in this genetic network. It's not central to the network. It's on the periphery of the network. Maybe this is a brand new gene, and I don't want to go into like how new genes form, but you get these new genes making new products, and maybe the product doesn't really have a lot of use to the organism, but it's making a product. Lots of things are made in genomes that don't really do anything. But there's a rearrangement in one of these areas of gene clusters, and because of the rearrangement, maybe this gene ends up becoming closer to another gene. And so now it's, let's draw it somewhere. Let's say it becomes associated with this particular gene here and changes something about it, right? Affects it. Well, now that gene just became potentially more important, right? It was on the periphery. And it was like, I'm just a side player. I'm not really critical to the life, to the ultimate success of this organism. But if I got a little bit closer, if I moved over here, and maybe if I just have a mutation and I make a slightly different product, all of a sudden my enzyme can now be used to do this, but it can also be used to tweak this thing over here. And now I'm actually affecting 2 biosynthetic pathways, or I'm involved in 2 gene regulatory networks instead of just 1. Now your presence might be a little more important. It's like the organism is like, oh, you're doing 2 things, right? You're affecting more of the organism. Maybe it's just a developmental thing at different times during development. All of a sudden I'm being used here, but I'm also being used later in life for something else. And then you have another mutation, right? Or you duplicate yourself. The other duplicant just becomes a side party and it does the same thing it was doing before, but your other duplicant has another mutation, and all of a sudden it's involved in, all of a sudden it's like closely related to a more core enzyme. Like it's working alongside something that's more central. You see how it's working its way in? This is kind of the way gene networks and developmental pathways are kind of visualized, is that you have a core network, and then how do you get more complicated organisms and more complicated things that organisms can do? Well, you keep adding on little things around the edge that tweak that core system and allow you to do something more, right? To make this digit a little bit different, do this other thing. And after a while, when that new change you've made becomes more important over time, because natural selection is looking at it going like, hey, we can actually use this, and this is actually, now we're working in an environment where we're using that particular product more than we were before. Then every time you have new changes to that particular thing, it becomes more and more and more important. I guess what you're saying is new genes, and this has been studied a lot. You look at a genome and you ask yourself, what are all the genes that are like varying the most and seem to have the highest rate of change and appear to be new because we don't see them in other species, we only see them in this species? Or in this case, there are genes that are only found in some individuals and not others. And they're those brand new genes that have a new function that no other individual in the species has. Those brand new genes usually don't do super important things. Okay, they're side players, but they start to work their way in. Over time, they're going to become more intricately linked, and as they become more linked, they're going to become more important. And as they become more important, the genome eventually will become more stable in that area. That's a long story about how that will occur, but that particular portion of the genome might become more stabilized because the organism selects for stability there, because it says, I can't play with this area anymore, right? I can't allow so much change because this has become an important set of genes. This now is a network. This is a pathway that I can't get rid of. And so therefore, I'm not going to allow it to change so much. In other words, it becomes like a core gene. And then you have a bunch of core genes. Well, what happens? You keep getting new genes and they become part of the system, right? You see how you can build complexity this way? Do you see how you can continually adapt a network? How a network can constantly be integrating new things and then finding new ways to do things? Because networks aren't just single lines. They're a web of interactions inside the organism that's constantly being worked and changed by natural selection, which is selecting which of these combinations work the best. Another way to look at it, and this might be a weird, maybe never have thought of this way, but here's another way of looking at a gene regulatory network. So this is a map of the dorsal ventral nerves in Drosophila. Gene regulatory network. I was just trying to remember what GRN stands for. The gene regulatory network for making and telling a newly developing embryo of Drosophila how to turn into having a dorsal and ventral side. Right, we know a ton about Drosophila and how it all develops. And what this is pointing out is all these different lines are the connections between these different regulatory things, regulatory genes and so forth. And so one thing is telling another to turn on, and then this thing turns on, this thing comes on, and it affects that. That makes a product which goes over here and then affects the connection here, which then turns on another gene and so forth. And this is all highly regulated. And this is all like, you have to do this in this particular order and this particular step, or you're going to mess up the embryo. Like from the very start, you got to have a very, very, very specific set of things. These are genes that, and regulatory networks that are shared among like all animals. Like all animals share a lot of these same connections. They're like deeply central, deep core type stuff that doesn't change very much at all. And so you're not playing around with this type of stuff, right? But with respect to like how large the wing is and what color it's going to be and how big the bristles are going to be on the side of a Drosophila's leg, those aren't nearly as important. So the developmental network there is going to have like additional little tweaks and changes that might vary between different species or between individuals within a species. And so the regulatory networks there are not going to be nearly as precise and as orderly because there's going to be a lot more variation. Now, I'm talking very generally here, just to give you a feel sort of for this network stuff. But I'm missing and not telling you a whole lot. And I also have a certain level of ignorance because this is so complicated, a bit out of my area, actually more than a bit out of my area. And there's going to be many other examples, but I come back to, I'm interested in genome structure and genome arrangements and orientations and understanding why we see the changes we do in those orientations. And that's really what this paper gets to, is how you can see and track deletions and inversions and changes to genomes, and then you can actually associate that with phenotypic changes to organisms, allowing them to adapt differently in their environments. And it's just cool to see that happening within a particular species. All right, I think that's as far as I want to go with this. Thanks for hanging with me and allowing me to explore this idea of where does new genetic information come from. And oh boy, I can tell I'm gonna keep going. All right, so this whole question of where genetic information comes from, I mean, there's the whole question of how you define information. But some people will say, well, all you've shown is that you can take previously existing information, like you have genes, and then you copy that information, and then maybe you do something different with it. But that doesn't explain where the original information came from. Okay, all right, granted. But it's still a very important point to be able to say that this organism couldn't express a certain biosynthetic pathway in its leaves, and its relatives, the other Arabidopsis, don't do that. But then when it duplicated a particular gene, it had mutations that changed its regulation, allowing it to be turned on in the leaves. And now those plants can do it in the leaves and the roots, and the other ones can only do it in the roots. That's a new feature for that plant. Right? That's a new characteristic. That's an adaptation, right? That allows it to do something in a different environment that the other plants aren't in, or if they are in, they can't do it as well. Right? So that is the origination of new characteristics. And if you don't want to call that new information, well, then don't call it new information, but don't say that new characteristics can't evolve from previously existing characteristics by adaptation through common descent. All right, well, actually modification, descent. Oh God, let me get this right. With descent, modification with descent. No, okay, descent with modification. I can't get those three words in the right order. Okay, my regulatory network is not working well in my brain at the moment. These examples show that organisms, through what are really mutations, right, which may or may not be, I'll say they're random. I mean, if you want to dispute that, that's fine. But they're still mutations. They're changes to the original genome. those particular combinations did not exist before. And if you copy a whole set of sequence and then you mutate it, you're creating new sets of A, T, C's, and G's in different orders that have never existed before. And so that's new, in my mind, that's new information. That's new information that exists that didn't exist before. Sure, it's a modification of previous information. And so you can just get into the chicken and the egg thing. Well, where did that information come from? Well, there was that particular gene network. That whole gene network I've been explaining that I said is not really that important of a network. It's possible that none of those genes existed in the ancestor to Arabidopsis, the genus. And that all of those genes have come into existence through copying other genes that were part of other networks. And then they themselves became a network doing a new biosynthetic pathway. Right? That's an even more radical change. It's not just changing one gene. It's not changing one regulatory region and so expressing in a different place. That's creating a whole new product that has a whole new phenotype. But you would still say that that was the result of copying previous information, right? But that's the whole point. I mean, information is pliable, changeable, adaptable. It can be changed and then reused used for new functions over time. And these can get increasingly complex over time. And that's the other argument that people say, is that complexity can't arise, right, from something simple. Well, if you only have 5 genes, and then later on you have 20 genes, and those 20 genes came from those 5 genes, you could say there isn't any new, all you've done is copy information. Well, you've copied information, but you have 20 genes, and now you're doing a whole lot of things you couldn't do before. You clearly have become more complicated. If you can make 3 more products that you couldn't make before, and natural selection adapts them to being used in different environments, that organism is now capable of doing something it could not do before. And so it has increased in complexity by virtue of just modifications of the genome that was there before, creating ever more complex regulatory networks. So I go back to here, right? If you're constantly adding a few new products that then begin to interact with some of the peripheral genetic networks, over time they'll become more and more integrated and become more important. And then what happens? The more of them will be added to those, and then more of them will be added to those. Now, sometimes there's deletions as well of systems. But it's pretty clear that genetic networks can grow and become more complex and more complicated over time, solely through the process of mutation and natural selection, selecting for networks that are adding advantages to the organism. That's my story of the origin of novel biosynthetic pathways, even within a single species. Thanks for listening. Talk to you later. Bye-bye.